everyone, this is SPN Kumar again. In this session we are going to discuss about biopharmaceutics and pharmacokinetics. So this subject has been incorporated into the syllabus in respect that if you become a formulation scientist, you should be well aware of the pharmacokinetic parameters and the pharmacodynamic parameters. So this subject biopharmaceutics, so let's go back to the title slide. So what is this subject and how this is going to help you in your graduation career, in your career in the industry? So this subject biopharmaceutics will give you an idea, about how the drug behaves in the body, the drug behavior or the drug characterization in the body. So there are two things which are occurring over here: what drug does to the body and what body does to the drug. So our body will absorb, distribute, metabolize and excrete the drug, whereas the drug shows its pharmacological profile. So it will be acting on the central nervous system, cardiovascular system, so whatnot. It will be acting on several physiological systems such that the therapeutic benefit can be obtained and we can reverse the what are the negative effects of the disease can be overcome with the help of this drug. So this subject biopharmaceutics is all about how the drug is behaving in the body, how it is coming in and going out of the body. All such things will be detailed in this topic, right? Such that by knowing this topic you can manipulate the physicochemical properties of the drug such that it shows a better therapeutic response and lesser adverse effects. So that's one point introduction about the biopharmaceutics. So let's move on to the first chapter and let us see what are the definitions. First of all, before getting into the insights of the subject, we should know a definition such that we can be well aware of this term getting repeated in your subject. So first thing is, what do you mean by biopharmaceutics? As I have said already, it studies all the factors which influences the drug in the systemic circulation. So whenever the drug enters into the body, whatever may be the factors which are involved, say for example there are certain factors like its solubility or its permeability. So all these factors are going to affect the behavior of the drug or the fate of the drug in the body. So study of all such factors is the principle of biopharmaceutics. Then what do you mean by bioavailability? Bioavailability is the amount, that is the rate and extent of drug that reaches the systemic circulation. For example, if you are taking a 500 mg of drug, not all drug is entering into your systemic circulation. Some amount of the drug is always being wasted, it is getting discarded. So say for example in the body only 450 mg or 300 mg of the drug is entering into the systemic circulation. Remaining all is getting wasted during the digestion or it is getting wasted during the process of absorption. So bioavailability is the actual figure how the drug is entering into the systemic circulation. So by knowing the bioavailability, we can reduce the wastage of the drug. For example, if you are taking an injection, all the 100% of the drug is entering into the circulation, whereas if you are taking a tablet, not 100 mg of not the 100% of drug is entering into systemic circulation, there is some wastage. So as a pharmacist you should be well aware of that wastages such that we can reduce the economical losses. So then what do you so in the bioavailability we have several factors like ADME. So your biopharmaceutics will deal with this absorption, how the drug is getting absorbed and how the drug is getting distributed in the body, right? So in the next slide we are going to see these parameters. So once a drug enters into the body, so there is this human body, so we are giving the drug molecule. So once the drug shows its effect, say for example let's plot a graph between time and plasma concentration. So the drug shows its effect, shows maximum effect, later on it gets reduced or it gets eliminated from the body. So this ascending phase is the absorption phase, whereas this descending slope is the elimination phase. So our liver and the kidneys are going to come into play and they are going to eliminate the drug away from the body. So this conversion is called as biotransformation. So whatever the active pharmaceutical ingredient is there, it will be acted upon by the enzymes and it will be converted to inactive form of the drug. So this inactive form of the drug will be eliminated away from the body. So if you know about this entry port and the exit port, you can easily estimate how much amount of drug will be there in the body, and you can provide maximum therapeutic benefit without having any adverse effects. So hence you need to know about biotransformation. So liver is the principal organ where biotransformation occurs, where drug will be converted from active form to inactive form. Then what do you mean by pharmacokinetics? Pharmacokinetics is, until now our hero is drug, what drug is doing to the body, but what our body does to the drug is coming under pharmacokinetics. So what body does to the drug? So body is absorbing the drug, distributing the drug, metabolizing the drug, and finally excreting the drug. So that is called as ADME. So for every drug there is a kinetics for this ADME. So some drugs they will follow first order kinetics, some drugs they will follow zero order kinetics. So remember a general rule. So zero order is concentration independent kinetics, whereas first order is concentration dependent kinetics. So we are going to see how this drug is getting absorbed. Is it following zero order or first order? How the drug is getting distributed? Is it following zero order, first order? So study of all these things will be encompassed under this biopharmaceutics. Next, so when we use this pharmacokinetic principles, pharmacokinetics in the sense the ADME principles, and you use it for clinical applications for treatment of the patient, that is called as clinical pharmacokinetics. How it is going to affect the patient's body will be study of clinical pharmacokinetics. So here is a simple flow chart how this ADME is occurring, as we have discussed earlier. So absorption is the process by which drug is entering into the body. Next, distribution. So once it enters into the body, say for example there is a tablet, it enters through this stomach, so then it will be entering into this large intestine and small intestine, where from the hepatic portal vein it will be coming to the liver, from the liver to the inferior vena cava, so to which it will reach the heart, and from heart it will be distributed all throughout the body. So this is a drug profile for a drug which is taken through oral route, that is a tablet or a capsule. So the drug is entering into this gastrointestinal system, but later on it is getting distributed all throughout the body. So study of such dispersal or disbursement of the drug is called as distribution. So by this distribution, say for example some drugs can undergo protein binding. So if it undergoes protein binding, the drug stays for prolonged period of time, it's not going to get eliminated easily. So here we need to give ample consideration towards this protein binding. Does your drug have protein binding or doesn't have protein binding? Next is metabolism. Once it gets distributed in the body, our protective mechanisms, the eliminating organs such as liver and kidney comes into play, and they are going to eliminate the drug from the body. So that is metabolism. So it metabolites. So first of all, the drug is a non-polar drug. This non-polar drug will be converted to a polar drug with the help of this metabolism, which is also called as biotransformation, and finally it is going to get eliminated from the body with the help of the kidneys. Once it is converted to a polar substance, our kidneys are good to go. It will handle easily all the hydrophilic substances. So that is ADME. And so let us see how the fate of the drug is there in the body. So we are administering the drug. Once it is administered, it undergoes disintegration. So this applies for a tablet. So tablet, it disintegrates into smaller granules. These granules will then get dissoluted in the physiological fluids, which is called as drug dissolution. From so for example, this is in the stomach, it is going to get absorbed in the small intestine through villi. Absorption is from villi of the small intestine, and it will be reaching the heart, where it gets distributed all throughout the body. So during distribution, if metabolism and excretion doesn't work, our drug will stay still in the body. So that is called as cumulative toxicity or accumulation. So hence that's the importance of liver and kidney. Say for example there is a patient who is suffering with hepatic failure or renal failure, then your drug will not be metabolized or excreted, then it will be accumulated in the body, which will result in toxicity. So as a clinical pharmacist, you should be aware that your dose should be reduced, especially if the patient is suffering with liver failure or renal compromise. So, and finally, some amount of the drug will reach the active site where it will be showing the pharmacological effects. So some effects may be beneficial, that is therapeutic effects. Some effects may be adverse effects or toxic effects. So ideally, for a good candidate of drug, therapeutic effects should be more and toxic effect should be less. Then it is called as an ideal drug candidate, and your effort should be to have maximum therapeutic effect and less toxic effects. So this is your target area. The role of biopharmacist is to enhance this therapeutic effects and minimize the toxic effects and minimize this accumulation. So let's move on to the next slide. Let us see how this whole scenario is working. So we are taking the drug. See, usually drugs may not have the intended physico-chemical properties. Drugs may not have the intended or desirable physico-chemical properties. So hence we are adding excipients such that a drug can be converted to a formulation like tablet or capsule or injection. So usually paracetamol, let's take the example of paracetamol. Paracetamol is utterly bitter in taste, so we can't give paracetamol directly to the body. So what we are doing, suppose there is a pediatric patient, so we are making a paracetamol syrup or paracetamol elixir where it is consisting of excipients such as sweetening agent, flavoring agents, coloring agents, which the final formulation is suitable for administration into the body. The final formulation, which has that better taste and a better color, better taste, can be administered into the body. So once it is prepared as a dosage form, some amount of drug may not be released, some amount of drug will be released by dissolution. Once it is administered, it will be releasing the drug from the formulation, and some amount of this drug, it is getting decomposed, metabolized or excreted. So that's why the concept of bioavailability comes into existence. So bioavailability is the amount of drug that reaches the systemic circulation. So some amount of drug is either not getting released or getting decomposed. Hence, even though if you are giving 100 mg of drug, not all 100 mg is entering into the body, only 70 mg or 60 mg is entering into the body. So as a pharmacist, we should minimize this wastage. You need to add excipients or manipulate this rate kinetics such that 100% of the drug is entering into the body. So then the drug which is undergoing dissolution will be present in the systemic circulation, and it will be reaching the site of action where it may show the pharmacological response or toxic effects, just like your previous slide. But here the point of formulation came into existence. So we can alter formulation. Say for example, tablet is not having bioavailability properly, we can prepare an injection which will have 100% bioavailability. So all these parameters can be taken into consideration to have a successful formulation. So this is a typical plot between time and plasma concentration. So let us take two different kinds of drugs. So let us suppose this curve which is having a spike, let it be A, and this curve which is more flattened, let it be B. So some drugs which after administration will increase its concentration in the body. Once the concentration is above the minimum effective concentration, it is going to have more therapeutic effect. But some drugs, they do not reach sufficient levels. Their plasma concentration is less than the minimum effective concentration. In such cases, it is not going to show the therapeutic effect. So ideally, your dose, whatever the dose which is present in the dosage form, it should be so designed that the dose is always exceeding the minimum effective concentration such that it crosses this barrier and shows the therapeutic effect. So in the given plot, A will be showing the therapeutic effect, whereas B is not showing the therapeutic effect because its dose is less than the minimum effective concentration to be established in the plasma systemic circulation. So that's how we can compare. So this is a bad formulation, B is bad and A is good. So your target should always be this good formulation where the dose is always above than the minimum effective concentration. But you should also be aware of the maximum line, which is called as maximum safe concentration. If your dose is above than the maximum safe concentration, it is going to result in adverse effects. So these 2 points should be borne in mind while we are preparing a formulation. Next. So now we understood that a drug is entering into the body So biopharmaceutics is all about drug entering into the body and drug exiting out of the body. So let us focus on the drug entry. So this is our drug entry. So how a drug can enter into the body? So there are the mechanisms of drug absorption. So we said that biopharmaceutics is study about ADME. The first one is absorption. So how the entry occurs into the body? So it can be by transcellular or intracellular transport, or paracellular transport, or vesicular transport. So these are all the three mechanisms by which the drug is entering into the body. So we can have a glance at all the mechanisms. So we will see each one in the coming slides. So let us see each mechanism in the slide. So first thing is the transcellular, paracellular and vesicular transport, all the three side headings compared at once in this diagram. So let us see the first one, so which is called as transcellular route. Trans in the sense the drug is transported across the cellular membrane. So this is your cell, your drug is passing into this cell membrane via the cell membrane. So this is a passive diffusion. So remember, passive is the mechanism which does not require energy. Whenever we say the word passive, it doesn't require energy. Then what is diffusion? Diffusion is a mechanism where solute flows from high concentration to low concentration. Diffusion is a process by which a solute travels from high concentration to low concentration. So in the transcellular transport, here the concentration of the drug will be more, and in the capillaries, that is in the blood vessels, the concentration will be low. So hence it moves from high concentration region to low concentration region. So this is a concentration gradient dependent process and it does not require energy, hence the name passive diffusion. Then what is carrier mediated transport? Suppose some drugs, they are non-polar in nature. Some drugs, they may be non-polar in nature. So in such cases, there should be a carrier molecule, just like a VIP treatment. So this carrier molecule is going to hold the drug, carry the drug across the membrane, and it is going to release onto the luminal side of these capillaries. So this is a carrier mediated transport. So that means drug alone cannot pass into the capillaries. It takes the help of the carrier, hence the name carrier mediated transport. Next is paracellular transport. So paracellular transport is the drug is traveling in between the passages of the cells, that is paracellular transport. So your drug is not passing into the cell, it is passing in between the cells. So para is the transport in between the cells. And finally vesicular transport. Vesicular in the sense vesicles will be formed. So the drug will be trapped in the pocket. So a pocket will be created in the cell. Your drug will be trapped in the pocket, travel across the cell and released onto the other side of the capillary. So these are three different ways. So we have three side headings here: transcellular or intracellular, paracellular, vesicular transport. So same thing is explained graphically or diagrammatically in a systematic manner in this slide, right? So now let us see each one. Let us see the characteristic features of each and every kind of transport. So it is transcellular or intracellular, which means we are discussing about this one, transcellular transport. So it is divided into two again: passive diffusion, carrier mediated transport. So let us see further details. So there is this passive transport. Passive in the sense, as I have said previously, it does not require energy. It moves according to the concentration gradient. So how this transcellular or intracellular transport occurs? So there are three steps involved. First is permeation of the drug into the GI epithelial cell membrane. So there is your cell, there is your drug molecule, it is going to permeate into the cell membrane, pass through the cell membrane. So next is movement across the intracellular space. Once it crosses the cell membrane, this is your cytosol, it dissolutes in the cytosol and passes to the other side of the cell membrane. So that's your second step. Third step is passing on to the lateral or basolateral membrane. So this is your starting membrane and this is your basolateral membrane. So your drug has to pass through both the borders. One is the starting superficial border and the basolateral membrane. Then it accesses the systemic circulation. Next is what are how this passive transport, how this transcellular transport can be further classified? It can be further classified as passive transport. As I said, it does not require energy. So how it can be further subclassified? It can be further subclassified as passive diffusion, which we have seen in the previous slide. So this is your passive diffusion. So the other one is pore transport. So we'll see shortly what is pore transport, ion pair transport are facilitated. So just remember the side headings. So PPIF, the mnemonic to remember all these processes are PPIF. So let us see the first one, passive diffusion. Passive diffusion in the sense it does not require energy. 90% of your drugs, majority of the drugs are passed by this passive diffusion, are flowing by this passive diffusion process. So how it is occurring? So in order to see the mathematics involved, it is following this modified Fick's first law of diffusion. So the amount of drug that passes across the membrane, amount of drug, dq, is the amount of drug, difference in concentration of the drug per a given interval of time, is directly proportional to the diffusion coefficient, area, partition coefficient, and inversely proportional to the thickness of the membrane. So whenever we have an equation, something in the numerator it is directly proportional, something in the denominator it is inversely proportional, and it is also directly proportional to the concentration difference in the GIT and the systemic circulation. So all the terms which are present in this equation are explained over here. Same thing which I have said previously: diffusion, area, partition coefficient, thickness, and the concentration difference. So pore transport. So what do you mean by pore transport? So this is also called as convective transport or bulk flow or filtration. So what happens is, suppose our cells will be having small cellular pores. So your solute is getting filtered from these cellular pores. So hence it is also called as filtration process or convective transport or bulk flow. So in order to pass through that small pore, its molecular weight should be less than 100 Daltons, because the pore is a very miniature size. Your particle size of the powder should also be very less, then only it can pass through that pore. Next, so only water soluble drugs can be followed by this ion pore transport. So examples include urea, water, sugars, etc. can travel by this pore transport, and some linear compounds can also pass through, especially their molecular weight should be 400 Daltons. So ideally we can say that for this kind of transport the size should be less than 400 Daltons. Preferably if it is closer to 100 Daltons, it can definitely pass through, but if it is 100 to 400, it has an intermediary flow by this ion pair, by this pore transport, right? So next, what is ion pair transport? So we'll see a diagram, then you can understand it better. So this is an ion pair transport. So it, for example, there is a drug which is positive in nature, its charge is positive, and there is a carrier whose charge is negative. We know that unlike poles attract each other, so this positive and negative poles are going to get attracted to each other. It is going to form a neutral ion pair complex. So there is a cation and there is an anion. It is forming a ion pair complex. So this complex, remember only one thing, only unionized drug will be absorbed. Unionized drug will be absorbed, whereas ionized drugs are not absorbed. So here, since both plus and minus got neutralized with each other, so we have